There are a number of human disorders that are inherited. And I typically have my AP Bio students learn about these human genetic disorders by conducting some research on an assigned disorder and then presenting their findings to their classmates. But I wanted to spend a little bit of time here in part 6 to go over with you 4 major categories of human genetic disorders, because part of your task when you're conducting research on your assigned disorder, part of your task is going to be to figure out which of these 4 categories your assigned disorder belongs to. And again, I'm categorizing these based on how they are passed down from parents to offspring. So the first of the 4 major categories is autosomal recessive. Okay, so obviously this would be a disorder that is caused by a recessive allele. So you're only going to show this trait, you're only going to have this disorder if you inherit 2 copies of the faulty allele, because again, it's a recessive allele, so it's only going to be shown, it's only going to be expressed if there are 2 copies present. So just to illustrate that, and I'm just using the letter A as a generic example, but the lowercase recessive allele is the one that causes the disorder. So notice little a would actually have the trait. But if a person has only inherited 1 copy of that allele, they would be normal. Now, we would describe that person as a carrier because they're heterozygous, but typically this person would not show symptoms, at least not any significant symptoms, and they probably would not even know that they were a carrier, until they produce a child someday with the condition. And then, of course, homozygous dominant would be normal and no chance of passing the allele down. Now let's talk about this word autosomal. It comes from the word autosome, and just as a reminder, all of your chromosomes that you inherited from your parents are autosomes except for the 23rd pair, which is either XX if you're female, or X and a Y if you're a male. Now this particular karyotype that we're looking at here is a human female, so there is no Y chromosome. But remember, if we say that a trait is autosomal, what that means is the gene that causes that trait is not carried on a sex chromosome. It's not carried on either an X or a Y chromosome, but it may be carried by any of these other chromosomes. And typically, if it's a well-known human genetic disorder, it's pretty easy to research and figure out which autosome carries the particular gene. So in your research, you may discover, well, the gene that causes this particular disease is carried on human chromosome number 7 or human chromosome number 14. Typically, you'll be able to figure out which chromosome it is. But if it's autosomal, it's going to be one of these first 22 pairs of chromosomes, and it is not carried on the X. So again, autosomal recessive, the gene is carried on one of the first 22 pairs of chromosomes, and you must inherit 2 copies in order to actually show the trait. Okay, let's take a look at the second major category, which is autosomal dominant. Now the first part is the same, autosomal. So remember, that means the gene that causes this particular disorder is carried on an autosome, which is any chromosome other than an X or a Y. But when we say it's a dominant trait, that means that the allele, the bad allele, the one that you don't want to have, is the dominant the capital A. And again, I'm just using A as a generic example. But because it's a dominant allele, it only takes one copy of that faulty allele to actually show the trait. That means that homozygous recessive would be normal. This is what you want to be. One copy of the faulty allele means you have the disease. And then homozygous dominant for these kinds of disorders is extremely rare, right? For obvious reasons, because the only way you could be homozygous dominant would be if both your mother and your father, who presumably are not related to each other, okay, if they both happen to have this enormously rare allele, that would be the only way you could get two copies. So that's just not going to happen very often. It's going to be extremely rare. And typically for autosomal dominant traits, homozygous dominant is usually fatal before birth. So if we're talking about a child that has been born, a child that has survived into its toddler years or childhood or early adulthood, there's no way that person could be homozygous dominant. So it really comes down to just two possibilities. For an autosomal dominant trait, you're either completely normal, little a, or you've got one copy of the faulty allele, and then you would have the disease. You're just not really going to ever see homozygous dominant for these sorts of traits. And one other thing here that's worth mentioning, it is possible for some autosomal dominant traits, it's possible sometimes that you have two parents, both mom and dad, are homozygous recessive. Mom is little a, dad is little a, and yet the kid is born big A, little a. You're like, how's that possible? That can't be. And it does happen. Sometimes it's a spontaneous mutation, where even though mom doesn't have a big A and dad doesn't have a big A, the child still can sometimes be born with one copy of big A just because the gene can mutate. And basically little a can mutate and become a big A. Not super common, but it does happen. It's worth mentioning because you may come across that in your research. Another thing that's worth mentioning, or I should say re-mentioning, because I've talked about this before in an earlier lesson, just because an allele is dominant does not mean that it's common. And that's a misconception that people have. I think most people just assume that dominant traits are going to be more common and recessive traits are going to be really, really rare. But sometimes the opposite is true. An allele can be dominant, meaning it only takes one copy of the allele to show the trait, and yet that allele can be extremely rare. I mentioned earlier a human condition called polydactyly, where a person is born with 6 fingers on a hand or 6 toes on a foot. And that's a dominant trait. That's a dominant allele that causes that. But it's an extremely rare allele. So your extra credit phrase that pays is this: dominant alleles can be extremely rare. Dominant alleles can be extremely rare. Remember, you got to be among the first 10 to email that phrase to me in order to get the extra credit. Okay, third category of human genetic disorders, sex-linked recessive. We've talked about this one fairly recently. Remember, if it's sex-linked, that means the allele that causes the disorder is carried on an X chromosome. So it's not autosomal. This is the opposite of autosomal. This is a gene that's carried on the X chromosome. And because women have 2 copies of that chromosome, a woman must inherit 2 copies of the recessive allele, I'm using hemophilia as an example here, a woman would have to be X little h in order to have hemophilia. But a male, a boy, only has to have one copy of little h to have hemophilia. Remember? So that's why these sex-linked recessive conditions are much, much, much more common amongst males, and they're very, very rare amongst females. So we talked about several examples: hemophilia, we talked about Duchenne's muscular dystrophy, red-green color blindness. Those were among the examples that I previously discussed. And finally, the fourth major category of human genetic disorder is referred to as aneuploidy. Strange word, aneuploidy. Now, technically, an aneuploid condition is not genetic because it's not caused by a gene. We're not talking about a recessive allele that you must inherit 2 copies of, and we're not talking about a dominant allele that you must only inherit 1 copy of. With aneuploidy, what is causing the problem, what is causing the human genetic disorder is not a gene, but it's simply the fact that the child has inherited an incorrect number of chromosomes. So you know how this is supposed to work. There should be 23 chromosomes in the sperm, 23 chromosomes in the egg, and that means the child, of course, will have 46, but it doesn't always work that way. Sometimes a sperm cell or egg cell can have an extra chromosome, 24 instead of 23. Or sometimes a sperm or egg can be missing a chromosome, 22 instead of 23. And so a child can be born with 45 chromosomes. That's aneuploidy. Or 47 chromosomes. That would also be aneuploidy. Aneuploidy is just a generic term that means having an incorrect number of chromosomes. Now, the cause of that is a phenomenon known as non-disjunction. We've discussed this before. This is something typically that goes awry. It's a mishap, a mishap that occurs usually during meiosis, during anaphase 1, when the homologous pairs are supposed to separate. Let's see if I've got here we go. So here's metaphase 1 of meiosis, where you have all your pairs of chromosomes lined up 2 by 2. Okay, and what is supposed to happen in anaphase 1 is each pair of chromosomes is supposed to separate and be pulled apart. But you see what's happening right here? Instead of these 2 chromosomes going their separate ways and ending up in separate cells, they both went to the left. Both of these chromosomes got pulled to the left. So that means we're going to have 1 too many chromosomes on this side, and we're going to have a missing chromosome on the other side. And so when meiosis is complete, if let's say we're making sperm cells here, half of the sperm made by this man are going to have 1 too many chromosomes, but the other half are going to be missing a chromosome. The failure of a homologous pair to separate is called non-disjunction. And sometimes non-disjunction occurs during mitosis, where you have a failure of sister chromatids to separate. But I think it's a little easier to kind of see how this occurs during meiosis, and that's probably the most typical way that this would happen. All right. So here's just to show you kind of visual evidence of what I'm talking about. This is a normal human female karyotype. So notice there's 2 chromosome number 1s, two number twos, two number threes. There's two of every single chromosome, and because this is a female, we even see two copies of the X chromosome. If I switch to a male karyotype, it looks almost identical, two of each type of chromosome, except only one copy of X, and then you see the Y chromosome. So this is a male, this is a normal male. And then let's compare this to the next picture. Now here once again we have a female, because notice there's two copies of X, and there's two copies of every single chromosome until we get to the 21st pair. Oh, it's not a pair. You notice instead of two copies of chromosome 21, there are three. Another name for this is trisomy 21. This is what the karyotype would look like from a person with Down syndrome. Notice there's a total of 47 chromosomes, and that's because of this extra copy of chromosome 21. So this is a great example of aneuploidy in humans. And this would typically be caused by either a sperm having an extra copy of chromosome 21 or an egg having an extra copy of chromosome 21. And typically with Down syndrome, it's more often going to be the egg. So you have an egg that's supposed to have one copy of chromosome 21, but it accidentally has two because of non-disjunction. And then here comes the sperm with a third copy. And so the child ends up with one extra copy. And that's an example of aneuploidy. So 4 major categories. And all of the human genetic disorders that I will be assigning to my students for them to conduct research on will fall into one of those 4 categories. So it'll either be autosomal recessive, autosomal dominant, sex-linked recessive, or it'll be aneuploidy. So I hope that's helpful. And hey, this is a pretty short lesson. It's only about 17 and a half minutes long. So I hope you enjoyed that. Don't forget the extra credit phrase that pays, and have a great day.